Welcome to all. Today we discuss about microbiology. This paper deals with various type of classification of microbes and also throws light on multivarious habitats of microbes and provides information about all the microbial cellular functions and various metabolic pathways in microbes. To impart knowledge on classification of microbes, this paper is also designed to provide knowledge on metabolic function and biochemical reaction going on inside the microbial cell. The organisms are not visible with naked eye and simply called microorganisms, and the world of this organism is called microbial world. The microbial world consisting of microorganisms like bacteria, fungi, virus, algae and protozoa. In this Pasteur experiment, Louis Pasteur believed that microorganisms are lived in air. 1908, Paul Ehrlich synthesized arsenical compound. 1924, Alexander Fleming found penicillin. In Berge's system of classification, the bacteria, fungus, virus and algae are systematically classified. Simpson's diversity indicates the term used to describe the number of individuals in a particular space. All microbes have a need for three things: carbon, energy and electrons. There are specific terms associated with the source. All organisms are carbon-based with macromolecules, proteins, carbohydrates, lipids. Nucleic acid having a fundamental core of carbon. On one hand, organisms can use reduced performed organic substances as a carbon source. These are the heterotrophs or other eaters. Alternatively, they can rely on carbon dioxide as a carbon source, reducing or fixing it is inorganic form of carbon into an organic molecule. These are autotrophs or self-feeders. For the microbial growth, the culture media has efficient nutrition for their growth. Culture media has macro and micronutrition. Culture media or growth media are solid, liquid or semi-solid substances that aid in the proliferation of bacterial populations. There are different types of culture media. They are simple media, complex media, synthetic media, enrichment media and differential media. Then 16S rRNA sequences. In 16S rRNA sequences, a type of rRNA involved in making the small subunit of prokaryotic ribosomes. These are components of 30S subunit positions. In 16S rRNA, S is the sedimentation coefficient, an index that indicates the macromolecule's downward velocity in the centrifugal field. The 16S rRNA gene is the sequence of DNA that corresponds to the rRNA encoding bacteria seen in the genome of bacteria. RFLP an enzymatic procedure for separation and identification of desired fragment of DNA. Using restriction endonuclease enzyme, fragments of DNA is obtained and the desired fragment is detected by using restriction probes. Then preservation technique. Preservation of culture the microbial cells by freezing, which can accomplished with either liquid nitrogen or employing cryogenic freezer. Then staining. In microbiology, staining can be defined as a technique which is used to enhance the contrast of biological specimen at the microscopic level. Then sterilization. Sterilization is defined as process of complete elimination or destruction of all forms of microbial life, which is carried out by various physical and chemical methods. There are different types of sterilization techniques. They are moist heat sterilization, dry heat sterilization, by radiation and pasteurization. Most of bacteria have haploid genome, a single chromosome consisting of a circular double-stranded DNA molecule. Conjugation is a method of transfer of genetic material from one bacteria to another. This method was proposed by Ladderbeck and Datum. They discovered that F factor can move between E. coli cells and proposed the concept of conjugation. Then transformation is the process DNA uptake by the bacteria from the surrounding environment. The cells that have ability to uptake DNA are known as competent cell. This process was reported in Streptococcus pneumonia by Griffith. Then metagenomics. Metagenomics is the study of the structure and function of entire nucleic acid sequence, isolated and analyzed from all organisms in a bulk sample. Thank you.